All right, so today's speaker is Chi-Chun Andy. So Chi-Chun is actually one of my friend back to college from entomology department, and he's doing PhD in University of Texas, Austin, and he's going to introduce some story about ants to us. So just give the time to Andy now.
All right.
Qin.
Thank you, Oscar. Thanks for the introduction. So today I have four things that I want to share with you. The main idea is to show you the embryo development of one of the leaf callers, one of the Antinians, and we also call it leaf callers. The species name is Atta texana. And since my childhood, I bet everybody watched Lion King before, and this is one clip from that movie. That scene was fascinating. That was actually the first time I knew, oh, there are some ants who will cut the leaves and bring them back. But at that moment, I didn't know what they were doing, or not even saying what is a social insects. So I'm going to show you what are the typical, like true social insects around us. Um, Termites, honeybees, ants, and wasps. So the typical features for social insects is that they have hierarchical level. For example, they have workers, they have queens. Sometimes they will have big soldiers, like a larger body size, and also all of the characters I mentioned, they're all females. For those sexual reproductive system social insects, they do have males. Okay, so how do we distinguish the male and the female social insects? Oh, by the way, termites, it's kind of awkward. The sexual determination for termites, it's different as honeybees, wasps, or ants. Okay, and we're not going to explore too much about termites today. Okay, so when we talk about the sex determination for eusocial insects, let's take ants as the example. This is the word that people will see. We call it haplodiploidy. So haploid means one set of chromosome, and diploid means like two sets of chromosome. Take us human being as the example. We are diploid. Okay, that means we have two sets of chromosome, and the way that human being decides the sex is based on X or Y chromosome. But the story is different in ant world. For the male ants, they actually only have one set of chromosome, which is haploid. And for the females, including queens or workers, they do have two sets of chromosome. So let's say if the egg got fertilized, that will become a future queen or a future worker, which is the female, right? If the egg didn't get fertilized successfully, can be a trophic egg or can be a male, right? So as I mentioned that Disney influenced me a lot since my childhood, but actually sometimes they make some mistakes. So this is the clip from The Bug's Life by Pixar Disney. The left-hand side, that's supposed to be a male worker in that movie. His name is Flick. And the right-hand side is the Princess Ada, a female virgin queen that Flick fall in love with. Okay. Um, the first thing Pixar made like wrongly is Flick can never be a male worker. There's no male worker. Okay. And the second, if we consider the real situation in the field, talking about Atta, this genus, Atta texana, the size of the queen is way bigger than the worker. So if we want to correct Pixar in the movie, it should be like this. So that's the queen and a worker, okay? And what's the real situation? Here is the Atta queen, and here is the worker, okay? So how do you decide? How does the egg decide to be a queen or a worker? Since both of them they are diploid, like fertilized eggs. It's still a mystery. Okay, so here I have a simple cartoon figure to show you question marks. Question marks on the slides means there's still something like the mechanism that we don't know. So back to 1990s, Diana Wheeler and other groups they thought juvenile hormone plays an important role at the late larval stage to determine whether that individual will become a future queen or just a worker. The more juvenile hormone got expressed, the higher possibility that individual will become a queen. Okay. And till 2015, another group, they figured out, oh, EGFR, which is the epidermal growth factor receptor gene, plays another important role during late larval stage, may involved in caste determination. But the other group from Canada, Ahad's lab, they published series of paper back to 2008 and 2010. And they brought a really interesting and important finding, as they thought the caste determination may be determined at a very, very early stage during oogenesis. Okay, oogenesis means before the egg being laid, i.e., the egg still in the ovary. Due to some gene regulation, their final character, like either queen or worker, will be determined. Okay. And how about the embryo development? Do people know anything about embryo development? The answer is no. Before I start to have my PhD, and that was actually quite surprising, because people study ants a lot. For example, like fire ants, like how to kill them, right? But or some interesting ecological concepts or behavioral studies. Like ants are one of the hottest species when you study insects that people will just use as a material. But surprisingly, we don't know too much during the egg stage or the larval stage or the pupa stage. Okay, people apparently care more about the later on stage, like the adult, like what's their behavior. So when I look back to those references, we left so many question marks. Okay, no matter like how many people study ants, we still don't know too much about egg stage. And there was the paper back to 2006, and they show some footage of the embryo development of two ants. The left-hand side, this one is Argentine ants, which is the huge pest, and this one is the Ferroni ants, also a pest, and it's quite common. We also have that in Taiwan. They in the whole paper, there's only 6 figures to show how does the embryo grow. But to me, it's not informative enough, right? Because I want to see like the start and the end. Based on the figure, people thought like maybe the ant's embryo development is similar to Drosophila, based on figure D. Okay, why do people think like that way? So there are 3 types of the embryo, the insect embryo, okay, based on the size of the germ-band. We call it insect fate map, okay. Here represents 3 types of the embryo, long, intermediate, and short, okay. The color part is the germ-band, which will become a future embryo, a future larva, okay. For the long-term band species, you will have the head, thorax, and abdomen part. Okay, if you are not too familiar with insects, that's the 3 of the majority part of the body of insects: head, thorax, and abdomen. Well, and wings and legs. For the intermediate germ-band species like beetles, at the beginning, once the egg being laid, you won't be able to see the abdomen part. You will see head part and part of the thorax, Okay, so does the short Germ-Band species like crickets. Okay, so I put a wasp here. For social insects, lots of scientists, they study honeybees and wasps, and they compare the Drosophila, one of the most famous model species that people study embryo development. They compare the wasp embryogenesis and the honeybee embryogenesis with Drosophila and figured out, oh, actually they're pretty similar. Those social insects, the wasps and the honeybees embryogenesis are pretty similar to Drosophila. So they didn't, people didn't pay enough attention to ants, and they just referred to based on whatever they knew. And they thought, oh, maybe ants, it's similar to honeybees or wasps because they belong to the same order. Okay, and today I'm going to show you that's not the truth. That's not the whole story. So 4 things I want to show you as the first. Well, I will talk about the ant embryogenesis, but I will involve 2 different species, except the Atta texana. I will also include Microcephalus smithi, and I will explain what is the high and lower agriculture mean later. That's the first part. And the second, I want to compare 2 types of eggs. One is laid by the foundress, one is laid by the mature queen. Foundress means the new daughter queen just fly out from the nest and have the mating flight and start to build their own kingdom, their own colony. Okay, the fresh queen. Okay, like the one you saw from the Pixar, The Bug's Life. That's the foundress. That's the virgin queen. And the third part, I'm going to use 2 genetic staining evidence to prove that the ant embryogenesis is not the same as honeybees or wasps. And the last part, that's kind of like a side project that I was lucky enough to figure out. We found out some new story about the tropic egg, and I will go deeper after. So here's the first part, the difference of embryogenesis between low and high agriculture ants. Here are the 2 species I include. The Atta texana, the leaf-collar ants, is the high-level agriculture based on the phylogeny tree, and I will show you in the next slide. And M. smithi is the low agriculture ants. Okay, and there are several features I want to point out to show how different these 2 ants are in different perspectives, like the ecology or even the reproductive system. First, Atta texana is sexual. They do have the male, but M. smithi, Microcephalus smithi, they're asexual. People also call them parthenogenesis. And the second feature is if you go to the fields, you will figure out there are different sizes of the workers of Atta texana. There are some small workers, there are some big workers, but for M. smithi, they are no difference between worker size. Okay, they're almost identical. Well, they're clonal, right? They're asexual, and so does the size. It's pretty similar. So what does the high and low agriculture mean? It's based on the position of the phylogeny tree. Okay, here is the low agriculture ant, the Microcephalus smithi, and Atta texana is the high agriculture. Okay, in the fields, the low agriculture ants not only use some dead leaf parts or roses or branches to cultivate their fungus, but also they collect caterpillar poops to grow the fungus. And the Atta texana, they basically cut the leaves and bring the leaves back to the colony, use those leaves as a nutrient to cultivate the fungus, grow the fungus, and they eat the fungus. So overall, we call them antennine ants. Okay, antennine ants means the ants will grow the fungus in no matter what perspectives, like in different ways, and they eat the fungus and they live the fungus. So they are farmers. Okay. So when we look at the life history for those 2 different antennine ants, they're on the 2 sides of the phylogeny phylogenetic tree, we may think, oh, they're so different based on their behavior, based on their ecology, even their morphology. They look so different. But surprisingly, they spend their egg stage, it's almost identical. So what does that mean? That means they spend most of the, they spend, both of them spend about 20 days for hatching. And how about those details during the embryogenesis, the features? Okay, so the next slide shows the first complete developmental table for the Atta texana. Okay, from day 1 to day 15, the eggs were from the foundresses, from those new daughter queens. Okay, so there are lots of information here, and I'm going to show you one by one, explain one by one. So the egg is about 300 micrometer, and day 2, you will see lots of nuclei showed up on the surface of the egg. We call it blastoderm. Okay, once you see that, the future embryo nuclei will start to gather to the posterior part of the egg and form a heart-shaped embryo. Okay, so first stage will be blastoderm, and the second stage will be the heart-shaped embryo. The third stage will be the elongation stage. Okay, when the heart-shaped embryo, the germ-band formed, the embryo will start to elongate and form like gradually day by day when it gets older. So the elongation stage is from day 4 to day 8. Okay, and insects, they are arthropods. They have exoskeleton, right? And they have different segments. So segmentation is the final step. That's when the embryo grows. So day 9, you can start to see those lobes forming, the segments forming, and till day 12, you can see compared with day 9, day 12, there's like more and more lobes adding up. Day 5, it's almost like lava. And then the next day, or within the same day, they will hatch. Okay, that's the overall concept of embryo development of Atta texana. How about Microcephalus smithi? It's pretty similar. Sorry, the footage is, some of the images are kind of foggy. That was the image I took almost 3 years ago, and we are recollecting the data for M. smithi. But on day 5, you can see the heart shape, the future embryo formed, and then the elongation happening, and then segmentation happening, just as same as what you saw from Atta texana. Okay, so if we look at those 4 stages of these two antennae ants, higher agriculture ants, Atta texana, and lower agriculture ants, Microcephalus smithi, shows really similar feature. Okay, so the first part, the conclusion is, even on the ecological or behavior perspectives, they're so different, but when we look at their embryogenesis, they're actually quite similar. All right. The second part is the differences between the Atta foundresses and the mature queen-laid eggs. So why is it interesting to compare these two? For a long time, people who study ants found out that for those foundresses laid eggs, they will usually hatch faster. Oh, sorry, not hatch faster only. For those foundresses, usually they will have the first broods of workers earlier than the mature colony. Okay, but for ants, there are different stages: egg stage, larva stage, pupa stage, and then worker. We don't know which stage got speed up, or every stage got speed up evenly. Okay, for a long time we just know, okay, for those new queens, they will have the first broods of workers faster than the regular queen, but we want to know more details. So since I have the expertise in studying and embryogenesis, the egg development, I examined the eggs laid by the foundresses the past summer, and we found out something interesting. So I hope you still have the concept for the whole developmental series in your mind of Atta texana. If we compare the eggs laid by those foundresses and the eggs laid by the mature queens on the same day, we can actually see the segmentation happens faster in the foundress laid eggs. Okay, this is on day 9. 3 mouse parts segments, the lobe formed, and the future thorax segments formed already on day 9 of the Atta new. Atta new means the foundresses laid eggs. Okay, but nothing happened except the future head lobe formed in the mature queen laid eggs. How about day 12? So if we compare day 12, we can see actually from the new Atta new, the foundresses laid eggs. Sorry, the phrase is so long. The foundresses laid eggs will form multiple segments at this stage, but not the mature queen laid eggs. Okay, but does that mean there's no gene expression involved in segmentation at this stage? No, that's wrong. So even there's no obvious morphological lobes formed from those mature queen laid eggs embryos at this stage. There's some conserved genes, for example, wingless here already has the expression. And I will go deeper for those gene expression later. At this moment, I want to point out that we firstly saw that actually during the egg stage, the accelerations got speed up already. Okay. How about the larval stage? How about the pupa stage? That we don't know yet, but they are different at the beginning. Okay, that means those foundresses laid eggs got speed up when the egg being laid. So those are the 4 stages: blastoism, heart shape, elongation, and segmentation, and a label at which date each stage happened. Okay, and what's the so why do we have to know that? Actually, that brought some interesting concept about maybe we should examine some aging genes involved in elongation or segmentation at this stage. Like we want to figure out what's the causation for that acceleration, for that speed up. It's quite interesting, and that will be our next step. All right. Okay. And the third part I want to explain is the Germ-Band type of the ants. Okay, recalled what we just discussed. The honeybees and the wasps, their Germ-Band type is the same as Drosophila. It's pretty similar to Drosophila. Drosophila is a fruit fly. Okay, they're long Germ-Band species, and I want to know whether ants has the similar Germ-Band or actually ants follow different development pattern. And when people study the Germ-Band, we use conserved genes. Okay, what does conserved genes means? So conserved genes means you can see the same genes showed up for similar purposes in different species. In the textbook example, people compare the Hox genes by flies, Drosophila, and a mouse. Okay, both of those 2 very different species, they have Hox genes, and they have similar principle in embryo development. Even the outcome is totally different. So the Hox genes expression involved in segmentation, formation, in insects, in flies, but in mammals, in mouse, it involves in bones, backbones, or somites formation. Like if you consider backbone, it's also kind of like segment-like concept. Okay. So the take-home message for the conserved genes means, okay, even no matter how different the species are, they share the similar genes involving similar principle. And how did I utilize those conserved genes in studying the Germ-Band? So here is quite a little bit complicated. I hope that can recall your high school biology or college biology. First, I want to say is from DNA to RNA to protein, there are different procedures, not procedures, sorry. So DNA will be transcribed to RNA and then will be translated to protein. Okay, that's how it works in the world, right? For sometimes, not every DNA will successfully be translated to protein. Sometimes it will just end at the transcription step, like they will stop at mRNA and no more protein will be made. Okay, and sometimes yes, you will see DNA, RNA, and then final products protein. Okay, for those genes, for those conserved genes which will stop at mRNA stage, we have to use in situ hybridization. And for the genes will successfully being translated to protein, we use immunohybridization. All right. So let me explain it slowly. For in situ hybridization, we have to make the RNA probe by using a dig labeled nucleotide. So dig label is just like a price tag. Okay. Attached to those nucleotides. Nucleotides are the code for your sequence for our DNA or RNA. Okay, and what are those codes? Those are ATCG and for DNA, AUCG for RNA. So we assemble those RNA probe with the dig labeled nucleotide. That's the first step, and the second step, we treat the sample with anti-dig antibody. So that's another concept. Back to your high school or college biology. What is the antigen? What is the antibody? So during spring, there's lots of pollen, right? And you will sneeze and you will have some immunoreaction. The reason why you will sneeze is because those pollens you sniff in, those are the antigen. And your body, you have your immune system. Your body will recognize that outrooter as a bad thing and start to make antibody. So when the next time you face the same antigen, like the pollen, your body will have enough antibody to make that reaction so you can sneeze those bad things which are pollens out, right? The bad things that is bad to your body. Okay, so this is the same idea. The dig label, the dig, is the site that the anti-dig antibody can bind to. Okay, it's like making different layers of sandwiches. Okay. And we also have the alkaline phosphatase attached to that antibody. And then we use chemicals, MBT and BCIP, to make the color. Okay, that's the reason why we can see with this successful binding between the dig label conjugated probe, the RNA probe we made with the dig, and the successful binding of the antibody dig, anti-dig antibody attached to it. We can, and then the next layer is the MBT BCIP. We can successfully see those colors. Okay. I hope I explained it clearly. So 3 layers. The first is the mRNA. Okay. The mRNA expression exists in the embryo somewhere, but how do we see that? right? We need to see some color, right? So we know where does it happen. And how do we know? First, we make the probe, the mRNA probe, to anneal with it, and then we treat them the antibody attached to another tag, and we use chemicals to trigger that colorization so we can see the color. When we see the color, we know where does the mRNA got expressed. Okay, that's the big story in short. That's in situ hybridization. Immunohybridization, same idea. We also treat them with different antibody. First layer is the antigen. You can consider as a protein. There are some binding sites on the protein that the antibody can recognize. Okay, so 2 layers in this picture. The first antibody will bind to that protein, right? And then we treat the sample with the second layer antibody, which attached to a fluorescent tag, so we can see the color after the tag got excited by the laser. Okay. Well, same idea. Okay. I hope you don't get confused at this moment. So for in situ hybridization, as for those expression stops at mRNA, right? Because there's no protein, so no protein can be produced. So there's no way you can use the immunohybridization to see the colorization. But for the immunohybridization, since you can easily make antibody attached to the protein, then we don't have to make the probe, right? And what do I want to see? There are 2 conserved genes involved in segmentation that you can see it in even really different species. So this is the example for grasshopper embryo development. Wingless is the first gene. Engrailed is the second gene. In each segment of the insect's embryo, wingless mRNA will be expressed at the anterior part of each segment. Engrailed protein will be translated at the posterior part of each segment. So in the real sample, you will see wingless, engrailed, wingless, engrailed, wingless, engrailed, just like the cartoon that I showed you. Okay, for the mouth part, for the thorax, for the abdominal segments. How about those expression in ants? Okay, and why is it important to see those 2 genes involved in ants? For Drosophila, for fruit fly, those segmentation gene expressions showed up in the same time. That's the typical feature for long Germ-Band species. So people study honeybee embryo development, they see the same pattern. Those bands showed up in the same time. Sometimes depends on the species. Sometimes it's like 14 stripes, sometimes like 16 stripes. Depends on whether it's a wasp or honeybees. Okay, but if you see all the bands showed up in the same time, that's the typical feature for a long Germ-Band species, but not for a short or intermediate Germ-Band species. That's the reason why I use those 2 genes to see whether ant embryo is similar to honeybees or not, right? So with the concept of the whole story of the ant development pattern, the embryogenesis, we can easily figure out when is the proper time for us to apply those probes, like the wingless or the engrailed antibody, right? We definitely don't want to apply those probes at a very early stage before any segments formed, right? Like the heart-shaped stage, no matter how hard we try, there's no way we can see the signal, right? We got to apply the probes or antibody at the right time, right? That's one of the important reasons why we have to know the developmental table first. So with that idea, with that concept, we know between day 9 to day 12, like when the segments starting to pop out, are the good timing for us to apply those conserved genes, probes, or antibody, okay, to see the signal expression. So for wingless on day 9, as you can see, there's no obvious stripes formed yet. But started by day 10, you can see 3 stripes over here. The top row and the bottom row, they are the same data, but I just use different way to show the bands to the audience. Right. And when the embryo gets older, more stripes are adding up. Okay, those are the stripes number. Day 9, you got 0. Day 10, we see 4. Between day 10 and day 11, we see 10. And until day 12, we got 12. Okay, so that's the evidence to prove that those segments, those stripes, are actually adding up. They didn't show up simultaneously. How about the ingrowed? Let me recall that wingless and ingrowed will be expressed at the same time. One is at the anterior, one is at the posterior of the segments, right? So we should be able to see the similar pattern when we examine the ingrowed gene expression, ingrowed protein. So the day 10 embryo, you can see there's only 8 stripes, okay? But when the embryo gets older, the stripes number get like adding up. So that's the second evidence to show, all right, actually, embryogenesis follow different development pattern as other social insects. Okay, so they're actually not long Germ-Band species. Okay, they're more similar to like Tribolium, like the beetles or the crickets. That's a really interesting and surprising finding. How about other social insects that is not similar to honeybees or wasps? Yes, we do find the evidence. There's another wasp show show the similar pattern, development pattern as the ants, which is either we can call it a short Germ-Band or intermediate Germ-Band development. So the green signals are the ingrowed protein, like the one you just showed. You can see when the embryo grow from A to H, you start to see the signal adding up. C, you only see one band, one. Yeah, you see, well, this is just the first band got formed. It's not the actual band yet. But D, you see like 3 stripes, and E, you got more stripes. Until H, like before, you see like 16 stripes, 14 to 16 stripes. Okay, and the red dots over here, those are another different gene called VASA. We're not going to explain too much. I'm not going to explain VASA today. Okay, so not just in ants, but also other social insects, different different wasps show the short germ or intermediate Germ-Band embryo development pattern. Right. So can we say that the ants has identical developmental pattern as crickets because they're not the same as flies or honeybees? No, we can't say that. I used the embryogenesis figure from 2 papers. First, we know, okay, based on the adding up stripes feature, ants are not like flies or honeybees. But the embryo, the Germ-Band formed completely. Okay, imagine the Germ-Band is the hot dog, and the yolk, the rest of the embryo is the bund. Okay, the ant embryo formed the hot dog first, like completely, and then segments formed, like gradually. But in the short Germ-Band species like crickets, you don't see the full hot dog first. You see part of the hot dog, and then more segments adding up. So when the embryo gets older, the hot dog the germ band gets longer and longer. Okay, in ants, you see the germ band formed completely, then segmentation. But in crickets, well, you don't see that for complete developed germ band. You see the segments and the germ band elongation like it happens. You see the segmentation adding up and the germ band elongation in the same time. Okay, so we think at this moment, based on our DAPI standing morphological data of the development table and 2 conserved gene expression data, the ants' embryogenesis is more like a mixture of long germ band and short germ band. Okay, and the next part will be the trophic egg. So trophic egg means the nutrient, okay? And for a long time, for those entomologists who study ants, trophic eggs are considered as one of the nutrients to those babies, okay? Here's the picture I took that the worker of Atta texana guards those eggs, her future sisters, in the fungus garden. Usually, the workers will chop off the trophic eggs and feed those larvae when the fungus start to grow. I mean, in the founding colony, right? In the founding colony, the queen only has small piece of fungus, so there's limited food resource. Therefore, the queen has to lay some trophic eggs and use those nutrients. Either the workers will use the trophic eggs to feed those babies, or before the first broods of workers come out, the queen will chop off those trophic eggs and feeds them as a nutrient. Feed to the first broods of baby as a nutrient. Okay, and the life history trait for a fungus growing ant is the queen lay eggs, and also she brought a small patch of the fungus from the mother colony and use her feces, poops, to cultivate the fungus. Meanwhile, take care of those broods and make them, help them to grow up successfully. And those workers, once she has enough workers, those workers will take care of her and also start to make the fungus garden be bigger, like this, right? And one interesting thing that I discovered, luckily, a few months ago, is we found a new type of trophic eggs, right? So when I first looked at that, I just thought, why the eggs, the morphology of the eggs is so different. It's so bizarre. It looks like 2 eggs attached to each other. And then I did multiple different sampling. I figured out actually here are the fertilized eggs, like here. And those 2 are the huge, massive trophic eggs that anneal to each other and even deformed. Okay, so I sampled, I got more samples from those foundresses, and we can clearly see that those foundresses they do lay trophic eggs, like those massive trophic eggs and the fertilized eggs in the same time. Okay, this is not the same you can see from the mature garden. For the mature garden that we have, 4 or 5 years old garden, those queens only lay fertilized eggs. So only during the foundering level, like when they start to establish the fungus garden, you can see those trophic eggs. And here is the cartoon figure to show 3 types of eggs. The first one is the fertilized eggs, like regular egg, the 2N egg, and the second is, we call it trophic A, and the third is the trophic B. Here's the actual figure for fertilized, trophic A, and trophic B. Okay? and I'll have more footage to show you how do I distinguish whether that egg is a trophic A or trophic B. So let's focus on trophic B first. One interesting feature for trophic B is the egg is just like a water balloon. It's so massive. And when you place it on the solid surface, it will deform, okay? And if you place it on the fungus garden, after 24 hours, it will be liquefied or deliquesced. Okay, what does that mean? Why is it important to see, like, why is it interesting? So trophic eggs, for a long time, people think or observe that either the new queen or the new workers will use them to feed the babies. But this is the first discovery to see that actually trophic B egg can also be one of the nutrients for the fungus garden. Okay. The fungus garden started with a small patch, like a really small piece, when the new queen fly out from their mother colony. And the fungus garden ants, the leaf-colored ants, they cannot live without fungus. No matter how many workers they got, once they lost the fungus, the whole colony will just die. Okay, so it's really important to make sure that small patch of the fungus the queen brought out from the mother colony can successfully be cultivated during the first or second month, right? Okay, so we hypothesize that the trophic B, the egg laid by the queen, has the evolutionary benefits to make sure the fungus garden can receive enough nutrient. This is another replication that we did the collection, and you can see after 2 hours in the first footage, you see 3 trophic B eggs. After 24 hours, we clearly see that the trophic eggs got liquefied. Okay, so would the egg be liquefied by the fungus only, or it will be liquefied just by itself? I mean, without the fungus. We're really interesting to know that. The next step we did is we put the trophic B egg on the cover slip in the close to 100% humidity environment. In the petri dish they created, after 24 hours, the egg will be deliquesced by itself. Okay, so it proves that this is a really nice co-evolutionary evidence to help the fungus, to help the queen, like on both parties, right? So the queen needs the fungus, and she will lay those trophic eggs to help her to maintain the fungus, to grow the fungus to be better. Okay, and the egg itself will deliquesce, so it will provide the nutrient or the water to that small fungus and help them to grow well at the beginning. All right, so we have more image to show the difference between the fertilized eggs, trophic A egg, and trophic B egg. For the fertilized eggs, usually the eggs will be guarded by the worker, and after 20 days, depends on the temperature, or 15 days, it will hatch to larvae, and will grow up to workers. And those fungus, the mycelium, cannot intrude to those eggs, cannot harm those eggs. Okay. On figure B, you can see the mycelium grow on the surface of the egg, but the egg is still pretty good, in a good status. And figure C, you can see the egg still like really like it's not damaged. But how about the other two trophic eggs, the trophic A and trophic B? Okay, for the trophic A egg, the mycelium will start to absorb the nutrient from inside and cause the dent, and eventually just sucked up all the nutrient by days, like after 13 days. And the trophic B egg, it will deliquesce by the cells. Okay, just like the way I explained to you, and how why it is important to help to cultivate the fungus garden. Okay, so besides the image evidence, I also fixed the trophic A egg to prove that we have two types of trophic egg. So for the fertilized egg, day 5, you can see the embryo start to grow, like during the elongation. This is the future head lobe. Here's the future body. But the trophic A egg, there's only a single nuclei, and nothing happens. And eventually, the fungus garden will absorb the nutrient. Okay. The last part of my talk will be, let's go back to those males. Okay. So I briefly explained how does the embryo grow in ants and the differences between lower and higher level agriculture ants. No matter how different they are, they show the similar developmental pattern of their embryos. And also, we've found out a new type of trophic eggs that people never discovered before, and we had the hypothesis that trophic eggs can also be the nutrient resource for the fungus garden, not just for the babies, the larvae. And one interesting we found out is, do you remember that I mentioned the haploid diploid of the insects? Some haploid eggs laid by the foundresses will become a male, okay? But usually that won't happen when the foundresses started to establish the colony, right? Because only female workers are useful that can help her to maintain everything. The male individual is totally useless at that stage. They do nothing. They basically just waste the energy, and also they are not helpful. Okay, ideally that won't happen, but we did find a weird nanotic male from those founding colony. Okay, and those males are really small. We call it nanotic. Nanotic means like lack of nutrient, and body size is really small. We found out that they have weird like fairy wings. The wings are not well developed, and they were just crawling around in the fungus garden. Once the pupa eclosion happened, like became an adult, the queen will kill it within 48 hours. Okay. Oops. Yeah. So here's the complete vision for the nanotic male of Atta texana. The morphology is totally different as the regular female workers. We don't know the queen can recognize the weird nanotic male by vision or by the smell. More studies we have to do. But at least one thing we can sure is during this stage, the egg, the larvae, or the pupa stage, here's the future nanotic male. During before pupa stage, the queen will still take care of them as the way the queen takes care of the workers. But once they become the weird crawling around male, they'll be killed, like they're literally ripped off by the queen. So that's also a new finding that we're excited to publish on Insect Social. um, of the behavior. So the queen knows those are not actual helpful workers, and they will just kill them. But the stories behind them is like how does she receive the signal? How does she know by vision or by other smell cues? Are really interesting to do more research. All right. I hope you guys don't fall asleep. It's a long talk, and overall, I finished the 4 parts. And thanks for your listening. And please ask me if you have any question. Thank you.
Okay, thanks for Andy's talk. Let's give in a pause first. No wonder me. Alright, so if you have any questions, please just go ahead and ask Andy.
How many people we have?
Now we have 6, including you.
Okay.
But
Sorry,
I think
the talk was like too long. I was starving.
no, I think you're fine. But your talk is pretty kind of professional, and even I don't understand. I would say half of your slides, because I don't study embryogenesis. Anyway, the development of embryos, right?
Yeah.
Right. So, but actually, I do have one question. Why do we care about so that question? So first, let me ask, what is the Germ-Band though?
Oh, so the Germ-Band means the embryo itself. Okay, so the embryo has the yolk and the future larva, and the Germ-Band just like the hot dog, and the yolk is like the bond. The overall, the two things we call it an egg, right?
Oh.
Yeah. So the future larva grow from the Germ-Band. Okay, start with the small heart and then start to grow bigger and bigger and then segments and then form the future larva. That's the Germ-Band.
And why do we need to care the difference between long and short Germ-Bands?
We actually don't have to care about that. So, well, no, that's not true. So that doesn't matter. That actually doesn't matter. Like, okay, yeah, it's long or short. But the reason why I want to study this is because the very first few slides I mentioned caste determination. So actually at the beginning, I really want to know what are the genes involved in caste determination in ants, if that actually happens during early stage. But without knowing the complete developmental table, I barely can do anything, right? I don't have the tool. For example, for like flies, people know how the embryo grows, so I can see different gene expression at different stage. With that knowledge, I can do some prediction, right? So if Ahab's theory is not saying it's wrong. I mean, if his theory is correct, we should be able to see some gene expression during the early embryo development.
Oh.
But how do I apply those probes? How do I apply those genes without knowing the developmental table? We can do nothing, right? So I got to study like how does the embryo grow first, okay? And surprisingly, we figured out, oh, okay, so actually they have different pattern like other social insects. So we divide them into different categories. So my finding just like to point out, ants, they are more like grasshoppers, like crickets, not like honeybees or wasps.
Okay, so that's just a step before you can move on to your further research.
That's the foundation.
Okay,
That's the foundation. Yes.
Okay,
But if you ask like why does long or short matter? No, that doesn't matter. It's just like a prescription, right?
Okay.
Mm-hmm.
Got it. All right. Are there any other questions?
Hi, I have two questions.
Cool.
Hi, I'm Jason. I'm Jason Hong. I'm like a physics postdoc. So I have no prior knowledge about the ant, and I have no limited knowledge about biology. So I have one pretty simple question.
Okay.
So you mentioned that for male ant, it only has like a one set of chromosome, and for female there are two, right?
Right.
So what determines that? So for human, we know that so you have two kind of sperm. So one is with X and one is Y, and that determines the sex of your birth. So what determines that in the ant colony?
Right. So the egg has a single set of chromosome. And if the egg got fertilized, the sperm will bring another set of chromosome into that egg, right? So if the egg got fertilized, it will be a 2N individual. Like it has two sets of chromosome. If
Uh-huh.
the egg didn't get fertilized, it will be a male, supposedly, from a mature colony.
Okay, I see.
Yeah. So
So
that's
so
the
even not fertilized egg can be hatched in ant egg.
theoretically yes, but there's some uh like you don't see male ants come out from the founding colony. Founding colony means the colony just started to grow. You only
Okay.
see the male ants come out from the mature colony.
Mm-hmm.
Yeah, so that's also a topic like people want to study the reason why.
Okay.
No,
So
no
we don't
matter
know
from
why yet.
we don't know why yet.
Okay.
Yeah,
And okay, so I have another question. So you mentioned that for the foundress ant and the mature queen egg, they have a different hatch time. Um,
right.
right. So also I guess you show the data, but I don't remember you explain why that is the case, and is there any method we can understand why? So is it due to chemicals or due to some some biological processes?
Okay, so we don't know the real key factors involved in that acceleration. And
Hmm.
for now, we guess it's because of a methylation. So, all right, let's put it in the easy way. So when think about you're the new mom and you have to start your colony, and there's so many birds or frogs trying to eat you, and you got to face those pressures in the field and start like also make having babies and
Hmm.
start to build your nest. So you want
Hmm.
to do that ASAP, right? You want to do that as soon as possible.
Hmm.
Yeah, and that's the reason why we think that makes sense that those foundresses broods will grow faster,
Okay. Hmm.
right? But
I see.
what's the mechanism behind this? What's the reason for causing that speeding up process? We don't know.
Hmm. Okay.
Yeah,
Oh, I
and
also. Oh, sorry. Maybe
no,
you
it's
can.
okay.
So I
Yeah,
also heard that in human, it's actually the first birth son, it will have a higher probability for early birth. Is that I mean that might be a very far away, but is that something also similar?
oh, I have no idea. Really? I actually don't know.
Okay, yeah. So I guess you can check. I don't
Okay.
know whether it is. It's a legend. It's a rumor kind of thing. I don't know.
Okay. Okay. Cool. I will check it.
Mhm.
Wait, Jason, you mean the firstborn will be larger than the others?
It will be faster. The pregnancy period is shorter.
Ooh. In
I
what
guess
animal though?
it's more like the like the rate for early birth is higher.
Interesting.
Oh,
Yeah. I would check it.
Okay.
oh, I haven't heard about that as well. So,
I'm not very sure, so you should double check.
Oh yeah, but that's interesting and important. All right. So are there any other questions or other concerns or suggestions for ND? All right. If not, I'm going to stop recording this talk. Uh,